Thanks, Natasha, for the nice introduction. And thank you, Tuan, and the whole committee for inviting me, and also for August to run this whole show. This is, of course, a yearly event, and it's a really interesting annual event that I'm looking forward to. So in this meeting, which has been going from atoms to galaxies, so I thought it was a really interesting range of applications of optics. We are sort of on the small end. We're looking at biological systems and cells and nanometer scale systems. We are using photonics and lasers all the time in everything we do, but we're actually not building biosensors or developing methods to sense something in biology, but we basically want to find out how biology itself or how biological systems themselves are sensing. And in particular, we want to find out how they sense mechanical forces. And so that is what is meant by mechanosensory. Now, obviously, everybody has heard about this one system that, if I get my slides to go forward, is the sense of hearing. In the hearing system, obviously, sound waves get transmitted to fluid flows, and these fluid flows get detected by specialized cells. And these specialized cells are called hair cells in the inner ear of the mammalian ear. And the cell is down here, and the cell has these so-called stereocilia, these rods that are sticking out of the top of the cell. And the fluid flow goes laterally across this bundle of hairs. And they're actual channels, mechanosensitive channels, that are located. It's not so clear exactly where they all are, but they're sort of either on top of this stereocilium or on the side of this stereocilium or both. And those channels are opened when the fluid flow is shearing this whole bundle. So that's introducing the sort of the final or the ultimate element that is actually sensing or converting the mechanical stimulation into an electrical signal, because of course these sensory cells are connected to neurons, and these neurons signal the activation and the influx of ions to the brain. In general, mechanosensitive or mechanosensation and mechanosensitive channels are in charge of many sensory processes, which include touch, hearing, of course, but also balance, pain, cardiovascular regulation, and also, and that's something I will focus on in this talk, a process called proprioception. And that is a process that provides feedback to the brain about the motion and the position of your muscles, basically, and of your limbs. There's a lot of work done, and a lot is known about the processes that are involved in this sensation, amplification mechanisms, adaptation mechanisms, and so forth. There's many channels, many families of channels known in organisms all the way from bacteria to humans. We're working on bacteria as well, but I will talk today about Drosophila larvae, to be precise. And obviously, Drosophila is one of the favorite model systems in biology, and it is very useful to work, or it's very good to work on, because there's a lot of mutants available, and of course the whole genome is sequenced and so forth. So it's a good lab animal to work with. To briefly come back to proprioception, so what is of course important when an organism wants to control its motion, its locomotion, whatever it's doing, or we moving our hands, the brain has to give the signal to the muscles, but of course the brain has to have feedback. It has to know what the muscles are doing at any given moment in time. And that is done by so-called spindle organs in our muscles. So there's sensors embedded in the muscle tissue that generate action potentials and basically have neuronal connections back to the brain that close the feedback loop. And that's how you can control your motions. You can close your eyes and still touch your ears. So obviously the eye can also help, but even without your eyes, you have this inbuilt feedback. And this is also true in insects. And in Drosophila, for example, there's again many places where mechanosensing is important, but the two prominent ones are in the adult fly, the antenna system that has to do with sound detection and airflow detection. That is on the head of the fly, and it's done by a so-called chordotonal organ. The whole organ in this case is called Johnson's organ, but the sensors inside this Johnson's organ are so-called chordotonal organs. The same or very similar chordotonal organs are also at work in the larva, and in the larva they serve mostly proprioceptory functions. And this is a larva here with its segments, and it's millimeters long, And in the next slide, you can see it in action. So here you have a larva that's trying to crawl. It's kind of stuck, so it can't get away. But you can see how the body segments are moving. The larva is extending and contracting. And what's highlighted here by fluorescent labeling are these so-called chordotonal organs, pentascolopedal chordotonal organs, in 2 in each segment of the larva. And these organs are stretch sensors. So you can see when we focus on one of them, while the larva is trying to move, you can see its length changes. So there's not much bending going on, but mostly when the muscles contract, you see a length change in that organ. So that's what the function of this organ is. It's called chordotonal organ because it looks like a chord of a musical instrument. So it actually is under tension, which we'll see in a moment. The anatomy of this organ is very stereotypical. So there are different types. There's the pentascolopedal organs that have 5 groups of cells, but there's also ones with a single group of cells and 2 and so forth. And they're actually occurring across many different types of insects. One of these components of the system is made out of 5 types of cells. Actually, I should say the attachment points on the outside attach to the cuticle, so the outer shell of the larva, and the muscles are kind of disconnected from them. The muscles are contracting and expanding and so forth, moving the cell around, moving the larva around, but the motion is sensed via the distortion of the cuticle. Then from this attachment cell on this side come so-called just connector cells, ligament cells. And here the blue ones are the neurons that then, of course, via axons communicate with the central nervous system. The sensory part where the channels are sitting is up here at the end of these sensory dendrites. And then come these very long cap cells. And you'll hear much more about these cap cells because they have very interesting material properties. But they're basically attachment cells as well, but they're sort of like as a rubber band to create the tension in the organ. So that's what I show you in our experiments. The inner part here, just to focus on that part where the sensing takes place, on the right-hand side are the neurons, and on the left-hand side are the cap cells. And this part is a so-called sensory dendrite. And this is where the channels are located, the mechanosensory channels, and where calcium influx happens. And in the end, of course, when the cell gets depolarized, the action potential is generated in the body of the neuron. Now, we were first interested in the mechanical properties because it's a sensory machinery. Of course, we would like to know what are the elastic properties and what are just the passive mechanical properties of this organ. Now, when you want to ask that question, you of course also want to know what are the proteins or what are the mechanically relevant structural elements in this machinery. And we know, or it was known before, that there's a lot of actin, which is a biopolymer that's also important in muscle contraction, both in the cap cells. So you can see it here labeling the cap cells, or you can see actin labeling in the cap cells. And you can see it here very brightly in the so-called scallopale cells, which were these shells that were around the sensory part. There's some in the other cells, but not so much. So mostly we'll focus on the cap cells. Here you see some fragments of muscle cells. So here's striated muscles, sort of the typical appearance of a muscle cell when you label actin. Now, in the cap cells, there's also tubulin. Tubulin is, for example, what you have in axons and also in dendrites and nerve cells. So it's a fairly rigid, elongated polymer that also, for example, makes the mitotic spindle in dividing cells. And that's a candidate for mechanical properties. But there's, as in many tissues, there's also extracellular matrix. Now, fly Drosophila only has one collagen type, collagen 4, but it also has a collagen-related molecule that's called pericardin. And it turns out pericardin is important because it makes a shell around around these cells, around the cap cells in particular. And we'll see in a moment that that is actually in charge of a lot of the mechanical properties. So these are the candidates for creating the mechanical stiffness and elasticity. Now, we did some super resolution imaging. This was actually before I moved my lab to Duke. I was in Göttingen, and of course, that's where Stefan Hell's group is, and the laser center in Göttingen with Alex Egner, the director, have a lot of the instrumentation that is developed up the hill in Stefan Hell's group. And so we were using the 3D STET microscopy that they have, the state of the art, to resolve the structure of actin in these cap cells. And what's intriguing is that the actin forms these sort of pellets that almost look like a braided structure, like a twisted kind of braid of these high intensity regions of actin that stretch along the whole length of the cap cells. And you'll see in a moment from the data that these cap cells are extremely stretchable. And of course, we were wondering why, how can that be? And the problem is that actin itself as a linear polymer, semi-flexible polymer, is not very stretchable. You can strain it by 5% or so, and then it breaks. These cap cells can be strained by 400%, as you see in a moment, and they don't break, or only break then. So we were curious about what the actin is doing. And obviously the structure, this kind of braided structure, has something to do with that. Oops, I had that problem yesterday. This tends to crash once in a while. Let me quickly get back to that. Sorry, I think I have to update my Microsoft. Go away. Let's see. I probably have to reshare my screen. Let's do this. And sorry, there's not much time, but I guess try to quickly go to where we were. So this was the stat image of one of these cap cells. Can you see the screen again? Is everything okay? Then we did a stat and zoomed into the sort of details of these pellet structures. And what we found actually is that this is, of course, the famous comparison. You always have to compare against confocal and show how much better super resolution microscopy is. But here it actually helps that you can see the connections between these bright pellets, and they consist of 4 actin filaments. These seem to be single actin filaments, and that's very stereotypical. Now, we don't quite know yet how the system works. We need probably even better microscopy or electron microscopy, but this was a very intriguing finding, and nobody has actually seen a structure like that before anywhere else. Now there's also myosin, which is the motor that also contracts our muscles. And if you do, in this case, it was 3D lattice light sheet microscopy here at Duke, you can see that there's sort of an interspersed arrangement of myosin and actin, which sort of looks like a muscle. So we expect there's some contractility also in these systems. Now, then the next thing we studied is the tension in these, the elasticity, the stretchability, but also the pretension that you find in these cap cells. And it turns out if you use a UV laser to cut these cap cells on the left-hand side in this movie here, this is cycling, of course, you see they snap back by about a factor of 2. So they're 100% pre-strained in their native configuration. And it turns out, to make the story short, we tried, of course, all the different structural elements that we suspected. We knocked out actin, we knocked out microtubules with no cortisol. Nothing happened really, but then we knocked out this collagen-like molecule that was forming the extracellular matrix, and lo and behold, there's no more retraction. So collagen, or this collagen-related molecule, is in charge of storing the elastic energy, basically, to produce that pretension. When that happens in the developmental process, we don't know yet, and nobody we asked knows that. So that's another avenue, another tangent we need to follow up on. But then we were also, of course, curious about the linear elastic properties, linear and nonlinear elastic properties. So here you see a glass needle experiment where we move with a piezo actuator. We move a glass needle, very fine needle that we can calibrate so we can measure forces, and push this organ sideways. Now, that's of course not the way it's usually being used in the cell, but of course we can easily elongate this group of cells, now 5 cells of this whole organ, and we can calculate from that, from the angles and the force we measure, we can calculate the tension in these cells. And it turns out they rupture only at something like 400% strain, which is comparable to silicon rubber. So it's quite extreme compared to other biological systems. The material has if you assume it's a homogeneous material, has a Young's modulus of about 300 kilopascal, which is between muscle and tendon. And it has a pretension of about 12 micronewtons stress, actually force, the pretension. And so that was not known before. But then I want to show you one more thing. We also did electrophysiology. And electrophysiology, of course, is good for recording action potentials from neurons. So what Chongling Guan, the postdoc who was doing these experiments, did, he was actually very experienced with this kind of experiment. So he managed to cut off the axons. This part you see here, that's the sort of stump of the axons coming from the neuronal somata. And he used a glass pipette, sucked the axons into the glass pipette, and then did patch clamp recording or current recordings. And at the same time, he had a second probe, mechanical probe, with which he could increase the tension on the organ and then record the action potentials and the frequency of spiking. So what you see here is in the top a wild type control organ, where you can see that when the force is increased, the tension is increased, the spike rate goes up, but it adapts pretty quickly. So within tens of milliseconds, it goes back to the normal rate. And when the tension is lost again, when the needle is moving back, you have the same thing. So you have a burst of spikes, and then there's adaptation again. So this is a detector that is differentiating the signal, the force. It's not giving you a constant signal, but it differentiates. It's a high-pass filter. Now, we were suspecting that myosin and actin in the cap cells has something to do with this adaptation mechanism. So we shut down the myosin partially with RNA interference, using RNA interference against the regulatory light chain of the myosin that's relevant, the non-muscle myosin 2. And you see the adaptation is less. It's not gone away, but it's less. But also, of course, this is not complete when we suppress the myosin. Interestingly, only when we increase the force, the tension, but not when we let go. So this proves that there's a feedback mechanism that basically turns on the myosin to counteract the tension that is changing in the cap cell. So if the tension goes up, myosin motors let go a little bit and decrease the tension. When the tension goes down, myosin motors pull in the rope basically and bring the tension back to the working point that the system desires. So I have to see how I'm doing on time here. I guess, yeah, almost out. So I have one more slide. But we try to put all these findings together in a coarse model for how the system works. Now, we don't know much yet about the receptor side, but it is known from biochemical or from cell biology studies that a motor channel protein called NOM-C, which is a TRP family channel, is in charge of the actual sensing. This is happening in the dendrite. This is mechanically connected to the cap cells. In the cap cells, you have sort of a big spring that is the collagen-like PRC molecule that is a passive spring. So it's a molecule that's outside of the cell. So there's no energy dissipation or any feedback that can change its spring stiffness or its force. But then there is, in parallel to that, the myosin generated forces and the actin network that can regulate and fine-tune the tension in the system. So let me leave you with this. So pericardin is in charge of that, and actin myosin is in charge of that feedback. And to allow a few minutes for questions, I'll put up the conclusions again in a moment, but let me just quickly mention the people who did all the work. This was started back before I moved here at the University of Göttingen, and Achintya Prahlad was a grad student who's by now a faculty member at Ashoka University in New Delhi in India, who started this. Chongling Guan is a postdoc who started in Göttingen and then came here and just left for UCL, who did most of the work I showed you today. And Kengo Nishi is now at UNC, is a postdoc involved in the various calculations. Martin Göpfert's group in Göttingen is a fly hearing group, and so we have collaborated with them and are still collaborating with them. And Xiaoshuan Jian has taken the baton now in my lab here at Duke and continues this work. And this was a collaboration with several people. Alex Egler already mentioned for the laser lab and the STAT experiments. Oliver Bäumchen's group, also in Göttingen now at the University of Bayreuth, has developed these needle experiments. And I didn't show you the optogenetics experiments that we did with Embel and Stefano De Renzi's group. But let me get to the conclusions, and hopefully we have maybe a second or two for questions if there are any. Thank you very much.